Okay, enzymes, as I'm sure we know, are biological catalysts, and they're made up of proteins. So the shape of an enzyme is absolutely crucial to its function. So we've seen with proteins, they do things like they form regions of alpha helix like that, and then you'll have a break from the alpha helix, and you'll have a bit more alpha helix, another break, and another bit of alpha helix, and so on. And then you have disulfide linkages between cysteine residues in the protein, and you might have some ionic interactions between a charged R group on one, minus charged COO minus, and NH3 plus on another R group of another residue, and that will hold the three-dimensional shape of the protein. And then, so very crude, you have a substrate, and that will fit into the active site of the enzyme, and the enzyme, it might bring, for example, the two reactants very close together and catalyze the reaction that way. Or it may be acid-base catalysis in the active site of the enzyme where one protons are donated and then taken away for whatever reason. But the most important thing is it's got to fit that active site very exactly. So let's have a think about this enzyme here, lactic dehydrogenase. So we've got, this is lactic acid here. It's 2-hydroxypropanoic acid. And it is reduced, the alcohol is reduced to the ketone here. This is pyruvic acid. Okay. So you can see you've got a carbonyl group there, you've got an alcohol group there. So it is it's an oxidation reaction, and the oxidizing agent, if you do biology, is actually NAD+. We won't worry about that though. But let's think about lactic acid. Now, if we have a look at lactic acid, I've drawn it again here. You can see it's got an asymmetric carbon atom, that central one there, which I put a red asterisk on. It's got 4 different groups around that carbon. Now, what I'm trying to show here is this black blob is the enzyme. And we've got one optical isomer on the left, so this is on the left, and this is a mirror, and we've got its mirror image on the right. So these are the two optical isomers of lactic acid. Now there's only one version of the lactic acid, the one I've drawn here, which can actually bind to the active site of the enzyme. Because if you see the carbox, for example, if you have the carboxylate group can fit into that red hole there, the hydroxy into that green hole, and the methyl into that blue hole. Okay. Now, if the other mirror image of lactic acid comes along here, you can see that it can't match up. Look, the carboxylic acid group and the methyl group are in the wrong places, and they can't bind to the surface of the enzyme. So we say the enzyme is stereo specific. It can only react with one optical isomer. Most enzymes are stereo specific. Both optical isomers, it can't react with them. So that's why, for example, in of all the amino acids, or 19 of the 20 naturally occurring amino acids can exist as 2 optical isomers. There's only one of them that's ever found in nature, because it's only one of them that the enzymes can actually recognize. The other one is the wrong shape, and it can't react. Okay, now if you look in the specification, we also need to know about certain drugs. Certain drugs, they work by inhibiting enzymes. Okay, and the way that they work is because the drug, it mimics the shape of the substrate. So it mimics the shape of the substrate, so it falls into the active site of the enzyme and competitively inhibits it. So if you've got the drug there, the substrate can't bind, the enzyme can't work. Okay, so there are a couple of sort of very well-known drugs which work in this way. Now, this is not a very well-known compound, but it's called, well, let's just have a look at this. It's called hydroxymethyl. The reason why it's called that is because look at this carbon here. You've got a hydroxy group and you've got a methyl group there. Hydroxymethyl gluteryl CoA. Don't worry what too much. CoA is an enzyme cofactor. Okay. What I would say is that if you look at this group here, it's very like an ester. The only difference is you've got a sulfur atom in there instead of an oxygen atom. It's an ester. Okay, and if you look at this in this reaction, what happens is that ester group becomes reduced to an alcohol group. So the carbon gets reduced to an alcohol. So it's a reduction. Okay. And I think the reducing agent, if you do biology, it's not NADH, it's NADPH. Okay. But the important thing about this enzyme is that it makes mevalonic acid, and mevalonic acid goes on many different steps. There's about 20 of them or something, and it eventually forms cholesterol. This enzyme is kind of like the gatekeeper to the whole process. The rate at which cholesterol is made is dependent upon how active this enzyme is. So certain drugs, and you've probably heard of them, called statins, they inhibit this enzyme. Okay, now when by inhibiting this enzyme, if you don't get the mevalonic acid, then you can't get the cholesterol at the end. So the aim of these drugs is to lower blood cholesterol. It stops your body making cholesterol. And of course, cholesterol involved in narrowing the arteries and heart, coronary heart disease. Okay, now another even better known enzyme inhibitor here is the enzymes are drugs which inhibit this enzyme cyclooxygenase. And I'll tell you what the name of the drugs are, and you'll probably, so the drugs that inhibit this are aspirin, ibuprofen, and ibuprofen is another classic. They're called non-steroidal anti-inflammatory drugs. Okay, and there's various other ones, like diclofenac, which is stronger than ibuprofen. But we see them as painkillers, but what they really do is they stop inflammation, and that can lead to they stop swelling and white blood cells being recruited to sites, and that's how they stop the pain, stop inflammation, stop pain. Right? How do they work? Well, there is this substance called arachidonic acid, you can see here, which is a carboxylic acid. It's 20 carbons long, and I've numbered them in a minute. Right, the enzyme is cyclooxygenase that reacts with oxygen, and it makes this compound, prostaglandin E2, and this substance, this general class of substances called prostaglandins, they promote inflammation. They do other things as well, but they promote inflammation. So if you can stop the production of prostaglandin E2, you can stop inflammation. That's how they work as pain relief. Right, I've just numbered the carbon atoms so you can see here. So I've actually numbered them backwards, which is a bit right. So really should call that number 1, but anyway, it doesn't matter. So you can see 5, 10, 15, 5, 10, 15, 20 there, and you've got 5, 10, 15, 20 there. Now what happens is you can see that the oxygen is going to react with carbon atoms number 10 and 12. So it reacts with these 2 carbon atoms, and it forms this is called a peroxide. Uh, so the peroxide link is when you get a single bond between two oxygens, like in hydrogen peroxide. So you can see, you can have a look at this molecule. You can see, for example, this double bond here at carbon 15, that's being conserved. This double bond has actually moved positions. It's not that one, it's moved one along. It's quite a complicated reaction there, but you can see how arachidonic acid gets turned into prostaglandin E2 by this enzyme. So you inhibit this enzyme, inhibit the production of prostaglandin E2, inhibit inflammation. So there's just, you don't need to remember either of those examples. The specification just says you just need to be aware that certain drugs work by inhibiting enzymes. So there's just two examples, and it's a good excuse to have a look at the skeletal formulae and things. Okay.